Hey everybody, I guess we're live. Welcome to another journal club presented by Life Extension Advocacy Foundation, LEAF, and Lifespan.io, where we host all of our projects. I'm Oliver Medvedik, coming into from New York City in the laboratory, the Kambar Laboratory Center for Biomedical Research, which I'm the director of at the Cooper Union for the Advancement of Science and Art. And this week's paper that we'll be doing is one that, well, it's exciting to me on two counts. One is it's a very recent paper dealing with the inhibition of senescence in the liver. And why would you want to inhibit senescence in the liver? Well, in general, most of you that have been following along with all of our journal clubs, senescence in general is thought of as a bad thing. We have cells that stop dividing as we grow old. And in the liver in particular, what excites me about this paper is that senescence appears to be a prime mover basically for acute liver toxicity or acute liver damage, and the inability of the liver, which normally is a highly regenerative organ, to not regenerate in cases of really severe acute damage that people suffer either through some sort of trauma or in many cases through overdoses, either accidental or otherwise, of certain toxins, particularly acetaminophen, which is found in Tylenol. So those of you who have Tylenol in your medicine cabinet, be very, very careful with it. It's an over-the-counter drug, but it actually is one of the main causes for young people, relatively young people, to get liver transplants. So if you got a bottle of maximum strength Tylenol, just be aware that if you pop one every 2 hours or so for that headache that won't go away, you'll pretty much be dead the next day. So just be very cautious with that stuff, even though it's over the counter. And that's what they looked at in this paper. They used an acetaminophen model of liver toxicity, and they wanted to see what would happen if they suppress senescence. And lo and behold, suppressing senescence actually is the key to promote liver regeneration in the models that they looked at here. And that basically, the exciting thing for me about this is that we've talked about how certain therapeutics can get to the market that are anti-aging therapeutics. And, clearly there's a bit of ways to go here in the United States to get the FDA to recognize aging as a disease or something that should or can be combated, which we all, believe it should be and can be based on all the scientific literature out there. But getting drugs to market is very, very difficult, even under the best of circumstances. And here we have a model where inhibiting senescence in the liver, may hit the market, sooner than we thought, as, through the application of, the indication of liver toxicity, because there's really right now no really good treatments for that. The paper suggests one possible treatment, an amino acid derivative, but it must be given within like 6 hours of toxicity, which is a very, very small window. And if that doesn't work, you need a liver transplant. So there's really nothing available out there. So to me, that suggests that this is a prime target, a prime area for a drug to be hitting the market very, very soon. Because thousands of people are affected by this every year. So that ties in with what we've talked about in previous journal clubs about how certain drugs can trickle into the market kind of through a back door. And one other, I'm just going to mention this briefly, that's indirectly related, but not inhibiting senescence, but getting rid of senescent cells. We talked about drugs that basically are senolytics. How would they get into the market? And clearly, senescent cells accumulate also through other toxic insults, such as chemotherapeutics. So getting rid of those cells in those types of indications would also be an inroad for drugs to start basically trickling in and then being used, by everybody else who's suffering through the ravages of aging. So with that, let's take a look at this paper. I'm going to pause and I'm going to share my screen. And this is it. Science Translational Medicine, 2018, Thomas G. Bird et al. With a ton of names and a whole slew of labs. TGF-beta inhibition restores the regenerative response in acute liver injury by suppressing paracrine senescence. So what does that mean? TGF-beta is a chemokine, which is basically it's a peptide. It's a class of peptides that are utilized by immune cells. They play a huge variety of roles, but in the context of this paper, and I think, Steve, if you want, you can chime in because you might have a better recollection of the Convoy paper that came out in 2015. That's kind of overlaps with the research here, although it's different labs altogether. But TGF-beta here is basically a driver of senescent cells. And there's a lot of downstream signaling events. But basically, what they look at here is a receptor, TGF-beta receptor 1, and then there's a ligand, which is the small peptide called TGF-beta, and there's different versions of it. And once it binds, it leads to downstream signaling. So there's a phosphorylation cascade. There's transcription factors called SMADs, which they look at in the paper, and that turns on all sorts of things downstream that have many effects, but in this case, leading to senescence or growth arrest. So what they show here is by basically screwing around with this pathway, by inhibiting it, you then block senescence. And not just block senescence, but by blocking the receptor, you basically also block the spread of senescence, because cells that are then senescent then lead to a cascade of basically release of TGF-beta ligand, and then that basically affects other cells. So here they basically talk about suppressing paracrine senescence, and paracrine means affecting other nearby cells, right? So you have the endocrine system, you have autocrine signaling, which is self-signaling, and paracrine signaling, which is kind of a localized kind of release of factors. And the factor here that they're looking at is TGF-beta, or I believe TGF-beta 1, which then binds to its ligand, and that leads to senescent cells kind of in the vicinity. So by suppressing all of that, they see that they can, I guess, remarkably induce regeneration. And this paper, the lead's a bit buried. For me, there's 6 meaty figures here that I don't know if we'll have time to go through all of them, but the more important 3 are the last 3, and the first 3 kind of set the stage for what happens in this acute liver toxicity. Anyway, I'm going to stop sharing this and go to our faces. Steve wants to chime in something. I know you've got a lot of insight onto this, which may or may not be your favorite molecule. I don't know. Maybe favorite molecule of the week, more likely.
You know me too well, but certainly it's in my top 10, and it has been for a while. Yeah, you're right. It was the main central player in the Convoy 2015 paper, where they demonstrated by inhibiting it, TGF-beta with ALK5, they managed to rejuvenate multiple tissues. I think there was muscle, and I think there was brain, and I think there was another one. I can't remember what it was now. But I think it might have been liver, actually, Oliver.
Mm.
I'll have another check, but it was at least 3. And from conversations I've had with Irina Convoy, they think it might be a mechanism that's common to most tissues, if not all. See, the key really is balance. Like in all things, TGF-beta does serve a function, but it's when it gets out of whack, when that balance is tipped, it goes from sort of being pro-youthful and helping maintain homeostasis, and then it flips over to a pro-aging profile, like so many of these cytokines. It's interesting to note that TGF-beta is also a common constituent of senescent cell SASP, which is the secreted signals, chemokines and cytokines and so on that come from senescent cells. So it's quite interesting. But TGF-beta is pretty much up there as a regulator of inflammatory responses, and it's also controlled by tumor necrotic factor alpha. So there we go. But we'll talk about that later, I think, because we're going to be going down the rabbit hole otherwise.
Yeah. So you mentioned SASP. So that's basically a form of kind of negative, or not. I don't want to use negative because that sounds like negative feedback, but a bad paracrine signaling, right? So that would be senescence associated secretory phenotype. So these things are secreting stuff. Some of the stuff is TGF-beta, or different versions of it, along with a whole bunch of other things that induce inflammation and induce other cells to senesce. And that's basically this aspect of SASP. This particular one molecule is what they're looking at inhibiting here, and it seems to the suggestion here is that inhibiting it can basically allow the liver to regenerate, which is very, very interesting. But anyway, so let's go back to paper here. Oops. Share my screen. All right. So, like I said, the first three figures basically kind of lay the groundwork as to what's going on. They're not really testing the means of inhibition yet. They're just showing what they're building models here and showing kind of what you see when you have a damaged liver. The first figure here is basically human liver necrosis. Dying cells causes acute hepatocellular senescence, right? So these are from liver transplantation for severe acetaminophen overdoses, right? So N equals 3. So these are human livers. So this is what you see when P21 expression. So this is immunohistochemistry, which is basically staining with antibodies. And a normal human liver here on the right. So all these little blue dots are probably nuclei, and all these little brown dots are P21s, which is a senescence marker. So basically, P21 is essentially a tumor suppressor. It basically blocks cellular growth or cellular proliferation or division, right? So it puts a brake on things, along with P16 and a lot of others. So cells have these brakes that keep themselves from proliferating, and normally they're there. Again, everything's in a balance. You don't want tumor suppressors to not be there. So, yes, you are removing them when you have acute toxicity so you can get your liver to regenerate, but you need them. Otherwise, you will have tumors growing all over the place. And if you are unfortunate to have, a mutation, then, you are very predisposed to getting tumors. So in this acetaminophen overdose and fulminant liver failure, this CV means central vein, and and they have these little dashes here, which I'm not a trained histologist, but so I'm just going by what they're saying here, which all of this are essentially, I guess this is your normal human liver, and you don't see all of these P21 being expressed at the periphery of this central vein. So you have these pockets, and they basically look under a high power microscopy, and I guess somebody does the counting, and this is degree of hepatocyte necrosis. So I don't exactly know how you call a cell necrotic. There's some visible way that you can define a necrotic cell, but basically, as the degree of hematocyte necrosis goes up, over here, I believe they do what do they do here? They count, basically, they do immunohistochemistry, and they count staining for P16, which is also builds up in senescent cells, prevents cells from proliferating, and that is a positive correlation, a direct correlation. And then you see an inverse correlation in the same kind of field with Ki67, which is basically a factor that's found in proliferating cells. So as cells, so that basically makes sense, right? So P21, P16 levels go up in cells that are based in areas that are necrotic, and proliferation factors go down. So that's probably not a big surprise, or maybe it is. I'm not sure if this was the first place where they noted this, at least in humans. So then we go into various mouse models, right? So here they want to recapitulate. In figure 2 and 3, there's a lot of stuff thrown here. you know, the one caveat I'm gonna give here is maybe they haven't. I haven't looked in through all of the supplemental figures. The one thing I wish here is that I had some wild type controls because I couldn't find them here. Because what they're doing here is mouse models of basically acute liver toxicity. You know, because obviously they're going to be experimenting on mice and not humans, and they're using two basically chemicals which damage cells. And particularly the liver is very sensitive. One of them is CCl4, which is carbon tetrachloride, and I believe it was used once in dry cleaning because it cleans your clothes very well and it evaporates, and I guess you probably don't want to be bathing in carbon tetrachloride. And acetaminophen, which is probably found in your medicine cabinet or first aid kit, right? And which will kill you if you take too much of it. We can talk about that later, but anyway, so here's basically what they've set up, these two models here. They have wild type mice and they show you what they see. So again, this is immunohistochemistry, CCl4, you know, carbon tetrachloride, acetaminophen, fields of view and cross sections of liver. This is how much they dose the mice. Day 1, day 2, day 4, and this is what they saw: early necrosis, peak senescence at day 2, ongoing regeneration, then recovery. So this wasn't enough to kill the mice, but enough to see damage. And again, these are cross sections, but you know, again, I don't know what the normal levels of P21 expression are in a mouse. I'm assuming they're pretty low, or the normal levels, you know, of, you know, in a mouse that's not basically dosed with this stuff. But here they're showing that, you know, all these brown dots are immunohistochemistry of P21 expression. These green dots of P21, what do we have here? Carbon tetrachloride. I guess this is immunohistochemistry. This is a different stain, I believe. I'm just going to scroll down here. There's a lot of, let's see, what do we have here? Myriad treatment. Expression of senescence marker P21, proliferation marker BRDU, bromodeoxyuridine, that's basically gets incorporated in DNA, magenta, so those are cells that are dividing, and hepatocyte-specific marker, which is this HNF4 alpha. So I don't know exactly what to make of this. This isn't really quantified. You do have P21 being expressed, right? Expression both in acetaminophen and in mice. You know, that was kind of my thing. Reading these immunohistochemistry, you know, slides is okay. I guess you're seeing P21 expression in the periphery around the central vein, where you have administration of these basically toxins, right? And then there's Ki67 expression, which is another proliferation marker. And again, it's really hard to, I mean, these are qualitative slides. So you've got somebody's hopefully blinded, right? So hopefully you do a blind analysis. So somebody who's counting doesn't know which is the control or which is not the control. And then you get these peaks. So these are days administration and P21 per field. So these are basically, they're counting all these P21 dots in an area and showing that you have, with carbon tetrachloride or acetaminophen, you have a certain peak happening 2 days administration of P21 expression, right? So basically, you know, they want to show in these mouse models that just like in human cells, you get a similar expression of factors and you have similar expression happening, you know, when you add these toxins. E is basically a plot. I believe it's a general gene set enrichment analysis showing, you know, acetaminophen injury to liver. So they're looking at basically gene expression profiles, comparing it to oncogene-induced senescence, showing that a similar set of genes, and it's probably in supplemental, is being upregulated in response to these stresses. So basically you're having senescence induced, right, in these liver cells. And what else do they look at here? So I'm jumping ahead to G, where they have basically P21 wild type or P21 knockout mice, and then seeing proliferation and regeneration. And clearly, if you have a knockout in P21, I just mentioned it's a tumor suppressor, so you should see much more proliferation. So P21 should be performing this role of inhibiting proliferation, and Ki67 is a proliferation factor, so wild type and P21 knockout. So this is after dosing with the acetaminophen. These little stars, I think, are the center area for necrosis. I guess this kind of whitish area is cells probably died or dying that are not taking up this stain, which actually may be hematoxylin eosin stain. But anyway, so here's P21 knockout, wild type. Hard to really see a difference, so somebody's got to really count this, and hopefully it's basically controlled for and blinded. So you can see that in P21 knockout, you have relatively more Ki67 positive cells. And here they do this serum ALT analysis, and that stands for serine alanine transaminase, which is basically released when the liver is damaged. So basically more of these levels that are there means that there's more damage occurring. And here they basically are trying to correlate tissue regeneration with inhibition of P21. So they're looking at here, so Ki67 hepatocytes per field. And I guess these are probably different mice because you have to take serum levels from mice, and mice that have, let's say, higher, so more proliferation that is occurring with greater damage. So you can see that cells are proliferating if you have P21 knocked out, even though so the more damage there is, the more proliferation is being induced to the cell. Basically, the liver is trying to regenerate, right? But you don't see this happening with a wild type control where there's P21. In fact, you see kind of less. You have an inverse correlation. So more damage that's occurring, there's less of these replicating cells that are occurring. More of them are senescent. So essentially, this is showing, stop sharing. You have this correlation between P21 through the various immunohistochemical analyses they've been doing, and the cell counts they've been taking, suggesting in mice that you have this correlation. Again, one of my pet peeves is maybe it's buried in there somewhere, but I'd like to see a normal, healthy liver as a control. And, I want to dig down a little deeper and to see how they do the actual counting, because, counting things under a microscope is tricky. You can easily bias yourself to count more or less. But I'm assuming that all the checks and balances have been done. So, I'm going to move along from that. Okay, so let me just scroll down here and get to the next figure. Share screen. This one might take me a little while to unpack because here they're now applying their mouse models. So they have basically mutant mice strains or knockout strains to see what would happen if they basically inhibit, inhibit aspects of autocrine signaling or senescence within a cell by basically turning up P53 expression and seeing if they can, seeing what would happen. So non-cell autonomous senescence is basically, so what we've been looking at in the previous figure is senescence within individual cells. And here they're trying to set up a model to see if they can measure senescence happening in cells that are next to other cells that are senescing. And here, I guess it gets a little bit tricky. This is a little, I think this needs a little more read-throughs to kind of go through for how they're exactly correlating paracrine senescence or, non-cell autonomous senescence. So here they have this model, right, where, so here, this model here where they have genetic induction of cells where you have cell autonomous senescence. Let me see if I can zoom in on this model here. Is there a way for me to zoom in on this? Where's my zoom feature? Nope. Oh, here it is. There, that's better. So you have basically, you have P53, basically P53 induction of downstream signaling and a secretion of TNF-beta 1. And then senescence transmission would then lead to a direct induction of P21, P16. So the way they're measuring paracrine senescence or non-cell autonomous senescence is cells that are expressing P16 and P21, but not P53, right? So basically, if cells are expressing P53 and P16 and P21, then they're counting that as autonomous. And non-cell autonomous would be cells that are not expressing P53, but through an indirect induction through a release of, later they'll go into the release of 2-minute Crescent factor beta 1, I believe, you have a downstream direct induction of P21 and P16, right? So bypassing the need for P53. And that's how they're going to basically then measure in these plots and say, okay, these are now paracrine cells, or these are now cells that have induced non-cell autonomously. I got to look at my pathways and see how, I guess, maybe there's people that are, watching right now who know more about the downstream signaling and whether or not you can have residual P16, P21 activity and basically a negative feedback that shuts off P53. But, and maybe you could mistake autocrine for paracrine in that way. I'm not sure, but that's the model. That's how they're teasing apart the two, right? So if that makes any sense. So they do in figure 3, they do these plots. They do these global genome analyses where they look at various, and there's a table that's buried in the supplemental that looks at gene expression, what happens when they look at various gene sets, and hours after acetaminophen, after Tylenol, and what sorts of gene sets are basically upregulated. So they do RNA-seq, and basically 12 hours after acetaminophen, you see TGF-beta signaling go up, inflammatory response. You have this oncogene-induced senescence response go up, and this is normalized, I believe, normalized to, I'm going to guess the wild type that's not been induced with acetaminophen, but okay. And that seems to go down a bit. I don't know why it peaks up a bit again. I'm not sure, but that seems to go up. And then slowly over time, you have these checkpoints that go in, which is basically a suppression of signaling, of replication. So your G2 to mitosis checkpoint, G2M checkpoint, mitotic spindle checkpoint, E2F targets, which are downstream transcriptional factor targets that lead to cell cycle arrest. All of these go up, right? So that's basically what we've seen earlier. So here we go to these mouse models that they have. And the mouse models that they use are, they knock out an inhibitor, MDM2 of P53. So these mice basically have a couple of different ways to do this. One way is that they use a drug that, oops, sorry, turn this off. They use a drug that essentially induces the expression of Cre recombinase that's specific in hepatocytes. So they have a promoter that's basically turning on this recombinase. It's an enzyme that, when it recognizes these binding sites, it'll cleave out whatever you want to cleave out or chop out. And this is what they're referring to as 85% recombination efficiency. So once you basically induce, and I believe they use a compound they call a xenobiotic, which is essentially a chemical like Tylenol that is not normally found in cells, that they can feed to the animal and induce this expression. I looked up the xenobiotic they used. I forgot what the name of it was. Again, I'd like to see a control, because you're feeding these animals this compound, and then you're seeing here massive P53 expression, which you're expecting, which I don't know what these arrows are really pointing to be honest with you. It's supposed to be pointing to P53 expression, but I'm assuming it's the brown stuff. So I don't know why these arrows are pointing to various areas of blue, because I'm assuming that's going to be that's going to be the immunohistochemical staining for P53. These arrows were getting a little bit confusing here. I don't know if there was maybe a little mislabeling here that was taking place. Because if we go through C, P53 accumulates in subpopulation of hepatocytes in the partial delta MDM2, and delta just means deletion, hep means hepatocyte-specific mouse model, where beta NF, which is that chemical, is given to AH, Cre plus MDM2 FLFL, and this is basically flocks. These are these little sites that the enzyme binds to get rid of MDM2. And then they do immunohistochemical staining assessed by confocal microscopy. Let me scroll down here to where they talk about what these arrows are. Gosh, where do they put the arrow stuff? Should put it right there so I can know what those arrows are pointing to. Well, anyway, the pink stuff is P53 expression. Green stuff is clearly P21 expression. So you seem to have cells that are just P53 and cells that are just P21, cells that are just P53 and P16. This white open arrow, I don't know if that's cells that are both expressing P53 and P16. I have to kind of go into the literature there and check. But you seem to have a lot of cells surrounded by P53 expressing cells that are expressing just either P16 or P21. Again, they mentioned that that's indicative of this non-cell autonomous senescence. Not 100% sure, but that's what they're saying that is a representation of. But anyway, again, I'd like to see also, since they're feeding these mice this chemical, I'd like to see, I'm assuming it's probably not going to be as severe, but what happens when you feed it this beta NF, or whatever that chemical is that they add into the water of the mice. Okay, I'm going to stop sharing for a moment here because there's a lot of talking. I don't know, are there any comments, any thoughts on the model? Anybody? Steve?
It's all quiet out there on social media, so they're probably thinking about the ramifications, aren't they, of regeneration?
Yeah, to be honest, when I read this paper, I was like, is there, and maybe we'll talk about this later, but is there a way to fast track this stuff? Because clearly, if your liver has just failed, you have 12 hours before you die. So or 24 hours. Well, whatever it is, it's a pretty short window. And later in the paper, they talk about, well, we need further trials, and I'm like, well, how fast are these trials going to take? 4 hours? Because if I need a liver, I'm not going to wait 4 years for a trial, right? Is there an ultra fast track for somebody who is about to drop dead due to liver toxicity? And they do mention that the drugs they're using actually are already drugs that are available, that already are used in for as cancer therapeutics. So these are these already have been vetted, I believe, through at least phase 1 and probably phase 2. And some of them, at least, I like to maybe phase 3. I think the ones that they used, at least one of the compounds they used later in the paper, it's been vetted probably all the way through and has been used clinically. So,
it's interesting that they're cancer drugs. I mean, it depends what they are, but it could be like, if it was like something like dasatinib, for example, that actually triggers apoptosis, as you know, in weakened cells and cancer cells, but it also tips senescent cells over the edge as well. So I'm wondering maybe if there's a similar situation that the cancer drugs are doing that in the liver, that they are tipping these near senescent cells over the edge, which then destroys and removes them, gets rid of the SASP, which of course contains TGF-beta. So are we potentially seeing a sort of senolytic action here, where the problem pro-inflammatory senescent cells are actually being destroyed, and that's what's alleviating and allowing the tissue, the liver tissue to bounce back, as it were. Could we be seeing something like that? Because I've got a pretty strong feeling that with senolytics in general, we're going to see upregulated tissue regeneration, just from the sheer fact that you're removing SASP and it certainly seems to be the case in animal models. And it's kind of exciting because it's about to enter human testing as well. But yeah, we definitely need a faster system. 4 years is a little bit too long to wait.
Yeah, especially if the numbers that they talk about are as high as they are, which is 3,000 people are needing new transplants annually in the United States. And the big bulk of it is, sadly caused by something that's entirely preventable, which is over-ingestion of acetaminophen, either accidentally or otherwise. And as I've learned, it is shockingly easy to accidentally kill yourself with Tylenol. If you basically look at the amount of milligrams you get in a bottle of Tylenol of acetaminophen, I think popping one every 2 hours will be enough to do it.
Yeah, well, Tylenol for everybody outside the US, I believe, is aspirin.
No, that's not aspirin. Aspirin is aspirin is it's different. It's not. It's
What is Tylenol then?
Tylenol is acetaminophen. Aspirin is it's a methylated version of salicylic acid. So I think So taking too much aspirin will lead to it'll corrode your stomach. It'll lead to you could
liver failure.
Also, I'm not so sure. Definitely hemorrhage. You could definitely that's why they give it sort of to kind of if you have a heart attack to help blood flow, to help thin the blood, right? They recommend chewing on a baby aspirin, but Tylenol is a different molecule altogether. It's aspirin was traditionally developed, basically willow bark, which is salicylic acid, and then they made it more stable by throwing a methyl group or something on it. So methyl salicylate, but acetaminophen is a different beast altogether.
It's paracetamol in the UK. I've just Googled it.
For Tylenol, right? Is paracetamol?
Yeah, Tylenol is paracetamol. So that's the other favourite that people take over here. I tend to say aspirin, but obviously there's a potential for bleeding with aspirin, so it's not something you want to make a habit of.
Yeah.
But then you shouldn't. At the end, the bottom line is, if you're having to pop a lot of painkillers all the time anyway, you really should probably go and see your doctor.
We'll probably prescribe more painkillers.
We'll probably prescribe some more painkillers. Yeah, probably morphine, opiates. But but it just goes to show that you do need to be very careful about when even over-the-counter things that are you'd think are fairly safe. You still need to be responsible and make sure that you're not overdosing, because it's very easily done. So,
well, so here's my take, and what kind of angers me is you got stuff like paracetamol, acetaminophen, Tylenol being over the counter, which is pretty nasty stuff, and the only thing it's really good for is, to my best of my knowledge, is a pain reliever, which there's so many other ways to basically relieve pain that won't lead to several thousand cases of liver toxicity a year that are basically induced by a product that's being, put into everybody's hands. And then you have stuff like metformin, which hasn't been known to kill anybody to the best of my knowledge, right? I think there was some acute toxicity of somebody who survived by ingesting like 73 grams of metformin. You don't need anywhere near that much of Tylenol to die. And metformin is under lock and key as a prescription drug. Why is that?
Yeah, it's the same. It's the same here. You can't get your hands on metformin at all unless you're there's a couple of ways, unless you're willing to risk importing it and getting it impounded,
right?
Or you're diabetic,
right? But I can go to my local, grocery here and get a truckload of Tylenol.
Yeah. Same here. I mean, we've got all sorts of things you'd probably go into a UK chemist and you'd be like, what the? Because there's so many things that are available here that are not available there, and vice versa. I mean, I understand that we have a lot of cough medicines here.
Yeah.
And they contain, I can't remember what the name of the particular compound is now, I forget. But there's a reason why they don't sell it in the US, because basically people use it to cook up and make meth.
Yeah.
But it's not a prevalent issue in the UK so much, but we sell like all the ingredients for meth.
Right.
You could just go to a chemist and go and buy it.
But just numbers wise, there's all of this lamentations occur, here of how do you save lives by doing this and that? I mean, I don't know. Maybe there's some medical doctor out there that can correct me if I'm wrong. But has Tylenol ever saved a human life? Has Tylenol ever been given to somebody to save their life? I don't think so. Tylenol is a pain reliever. Is there ever been an acute case of is there any indication out there where they're like, oh my God, give this person Tylenol and this will save this person's life? If not, then why is it still on the market? Why don't, if by just pulling paracetamol off the market, you basically get like 2,000 people off the liver transplant list every year, and they're lamenting that we have such a long waiting period for liver transplants, and it's caused mostly by Tylenol overdoses, which a big chunk is accidental because the dosage to kill yourself is so low, and Tylenol doesn't save lives, but it may relieve your headache. Why is it still on the market?
It's nonsensical, isn't it? It's I suppose it's a little bit like aspirin. I mean, it's really quite dangerous if you, well, anything is really.
No, but if it doesn't save lives, if there's been not a single documented, if it's proven to kill over 1,000 or 2,000, whatever that chunk of 3,000 people, which they say is the number one reason most people, at least in the United States, for getting liver transplants, is acetaminophen overdosing for a variety of reasons. Why the hell is it still on the market if it's not a life-saving drug?
Yep, I'm not aware of anybody being saved by paracetamol.
So I don't get it. Can an MD please chime in, phone us in and say, no, you're flat out wrong. If those 3,000 people that need liver transplants, if paracetamol wasn't on the market, we'd have 30,000 more deaths. Am I missing something here?
I'm going to say what I always say, follow the money, right?
Anyway, so stuff like that, and then having metformin under lock and key really gets my goat.
Yeah, I mean, there's really, really good evidence now for metformin might be beneficial for people who are pre-diabetic and stuff like that. It's getting very popular in life extension. I don't think it'll add much, but at least it doesn't lead to thousands of people ending up on liver transplant lists. Yeah, I'm pretty sure about that. I tend to avoid eating those like candy. Yeah. But there is a real issue. The other one over here is ibuprofen. Ibuprofen. Yeah, Advil. That's it, yeah. Oh, Advil. You see things I learned today, all these brand names. Name brands. It's Advil, and also you go to a drugstore and you say, well, I for one, I know it does, and I looked it up, does increase the incidence of perhaps, like hemorrhage, but at least for me, Advil works. It's nowhere near as toxic as Tylenol, and it does go under a lot of different brand names. So if you go into our corner grocery store here in the States, you'll find Advil, you'll find Motrin, you'll find a whole bunch of other. They're all the same thing. It's ibuprofen. Yeah, but you know, crazy differences. I mean, you guys can buy Valium over the counter. I mean, come on. I don't think we can buy Valium over the counter. We can buy. I thought you could. No, you can't buy. But you can buy other. You can buy what's that? Melatonin over the counter. I think you can get that here over the counter. I thought maybe I imagined that, or maybe it used to be the case, but I always thought you guys could buy Valium. I don't think so. No. Yeah, maybe we should allow that in the UK. It makes some people chill out. You know, there's a lot of angst at the moment here in the UK. I think everybody should just have some Valium. You know, calm down. Well, thank God marijuana is still illegal. Otherwise, we would all die, right? No. If it wasn't, everybody would just everyone would just chill out, and there'd be an outbreak of lethargy. Never get anything done. What would you rather have your kid do? Smoke a joint or eat a bottle of Tylenol? Quick, decide. Smoke a joint. Anyway,
I veered off slightly off topic. That was my rant against medical establishment and how things don't make any sense and how they could easily make sense. You want people off the liver donor list? Ban Tylenol. There, done. I just solved your problem. How you're going to do that? Well, I think that's the even bigger problem, and I don't have a solution for that. So, let me go back to paper here. Okay. So scrolling back here, back to the mouse models. So again, like I mentioned, MDM2 is a negative regulator of P53. Remove MDM2 and you have P53 levels go up. P53 levels go up, you induce senescence. You stop cells from replicating. That's what P53 is probably the most important. You know, if you haven't heard of it, you've probably been under a rock for the past several decades. But P53 is basically a little protein that's about 53 kilodaltons, hence protein 53. Not a fancy name, but it's basically mutated in pretty much most cancers, or half the cancers, or whatever the percentage is. It's pretty damn large. So P53 is important for maintaining genome integrity, and it turns on basically lots of other genes that are required to stop cells from replicating when they're cancerous. But, you know, again, too much of a good thing could be also bad. If the cells start to senesce, you know, probably don't want P53 overregulated. But for the most part, P53 is there to prevent you from dying of massive tumors growing all over your body. But here they can basically stop this autocrine and paracrine signaling by basically shutting down P50. Well, here what they're going to do is induce senescence in this model and see what happens by turning up P53 by blocking its inhibitor, right? Just had a brain seizure here, but by blocking its inhibitor, its regulator, and getting P53 induction to go up. They use another way to basically delete MDM2 in hepatocyte-specific way. They use this viral vector AAV8 and this Cre recombinase system, and I think they can titrate this. I don't know why they use these two systems. So they use basically a Cre recombinase system using a drug and using this vector, probably to alleviate concerns that maybe the drug's also causing some sort of toxicity as a control. And they mentioned partial delta MDM2, and this is where they start to kind of, you know, use this model to suggest that, you know, they don't want to knock out P53 and everything, but here they have 85% recombination efficiency up here. Here they want to basically add in a little bit of this molecule and probably a little bit of this viral vector to essentially turn off just, you know, not in every cell, but in some cells, MDM2, and cause P53 levels to go up. And then I guess that's would make it clear for them to see cells that are upregulating P21 that are adjacent to P53 upregulated cells. So basically, partial senescence. So here's their control. So P21 is in pink, P53 is in green, and DAPI is staining for DNA. And then they induce, so this is not adding, this is not basically knocking out MDM2. When they do, here in hepatocyte-specific cells, they have arrows pointing here. And gosh, they mentioned somewhere what these arrows were. You've got the paper somewhere, Steve. Somewhere buried in here is the legend for, oh, here it is, open arrow. I guess open arrow is this, maybe. Open arrow, cell autonomous senescence. Closed arrow, which I believe is this one, or is it that one? I think it's this one. Closed arrow is non-cell autonomous senescence. So an arrowhead is unaffected, which is this arrow. Okay, well, so nothing lighting up here except DAPI. This one here is supposed to be non-cell or cell autonomous, which is basically P53 only. I'm not sure which one this is pointing to. I think maybe this one, which is green, and P21 is purple, so that should be non-cell autonomous. So that should be, so basically you see ones that are P53, and I think this one may be a merged again, it's really hard to tell here if this is actually both P53 and P21, and they would consider that to be, but they would, anything with P53 expression, they would consider to be cell autonomous. So that's basically their criteria here. You can complain about it if you wish, but that is their model, that is their criteria. E, I got to remind myself what they're doing here. They're doing a kind of a transplantation with cells that are GFP positive. So here they partially delete MDM2 using this chemical. That's another way to induce Cre recombinase. So again, you get senescence. They add in this transplant, and then they do repeated after the transplant, after day 14, repeated ablation of MDM2. So basically, they're inducing senescence. And this is a sham transplant control. So no transplant here, DAPI, lots of P21 cells. And what are they trying to show here? GFP cell transplant. So, okay, they put in GFP positive cells. Nearby, you've got these senescent cells, you've got DAPI stained cells. And what do you have here? These GFP cell transplants are, well, I guess this is supposed to show that these are in the same cell, I guess, that these GFP cell transplants are themselves being upregulating P21, which, would be an important, really important. So I think this is a pretty important figure. I don't know if they have this really quantified, which I would really love to see this quantified. Maybe it's in somewhere inside the supplemental. But, if you're adding in transplanted cells into this mouse model that doesn't have, and these cells are normal cells, and you're seeing P21 expression, that would be, that would be a clear cut, much more clear cut to me, that you have paracrine signaling taking place that's inducing senescence. I would like to see GFP cell transplants also being put into cells that are not induced with this chemical, right? So you should see GFP cell transplants that are just GFP if you transplant them into a mouse model that is not being ablated from MDM2, right? You should see less P21 expression in those transplanted cells, meaning less paracrine signaling. I don't know if they have that experiment or that figure. I think that's a pretty important experiment. Maybe they do. My apologies if they have that in there somewhere, but I don't recall seeing that. Because I think that would be really key for me to kind of cement the case for this paracrine signaling that's taking place, at least the visualization thereof. Okay, so we'll just quickly move on to F, G, and H here. So here they have another model where they have this Cre recombinase system that is able to make a constitutively upregulated TGF-beta receptor 1. So basically, TGF-beta 1 receptor is what is going to be necessary for downstream signaling and induction of paracrine signaling of senescence in cells like these GFP transplant cells. So if you've got this stuff upregulated, if TGF-beta receptor 1 is always on and always signaling, then these cells should be expressing P21 and other markers of senescence, right? So that's not a surprise, and they do. So here's a control, P21, immunohistochemistry, TGF-beta R1. You have expression all over the place. And what do they do here? So in figure G, again, they do this. They do the same thing. They see so Tom is a reporter. I've got all these cute names. Tom, in case you're wondering, does not stand for, isn't Thomas, it's tomato. And it is red fluorescence protein. And these cells, the RFP should be stained. It's basically fusion protein. I think it's fused to TGF-beta receptor 1, or it's basically co-expressed with TGF-beta receptor 1 that's constitutively active to show that these cells actually are making it, right? So the RFP so here's your RFP cell, P21 is green. So you have some cells here that are actually just green, some cells here that are just red, and some cells that are both. So probably this would be your, these two I think would be your cell autonomous, and this one would be your non-cell autonomous. It's so confusing to hear non-cell autonomous. Just these are your paracrine-induced cells, basically. And over here is where we kind of get to the kind of the cool stuff. So this jargony number here, SB525334. God, you know, if you can come up with a name like Tom or Tomato, can you come up with a better name than SB525334? Yeah. It's just like a gamish of all sorts of names and terminology here. It's like some things are cute and some things are just frightening.
SB what? Wasn't he in Star Wars?
Yeah, SB525334. I think that's a droid.
Yeah.
Droid number.
I think that one's got a bad motivator.
The Jawas are taking it apart right now. Okay. We should stop, otherwise we're just going to confuse the hell out of even more people. So figure H here is so partial dosing, again, by essentially what they're doing here is cranking up the p53 levels in these hepatocytes, and then they're adding in this chemical compound that is binds to to TGF-beta receptor 1, and it basically blocks it from binding to ligand. I think that's what it's doing. It's basically a competitive inhibitor. If, you know, might not be a competitive inhibitor. Again, somebody chime in if they know exactly how SB525334 works. But I think it's a small molecule, and that's how it functions. So here's your control. Here's your treated. I don't know exactly when that's when they treated it. They kept dosing it. And per field means, so somebody, again, is counting cells. So non-cell autonomous senescent hepatocytes per field. So I'm assuming those are the cells that are just expressing P21. And the numbers go down per field, you know. Again, I'm assuming somebody's doing this blinded, or they have an automated procedure, because again, you know, counting cells under a microscope can drive you nuts, as I've done before, and you can easily bias yourself. But I'm assuming these numbers are, you know, hold fast. So this compound is basically, you know, I mean, is according to their model, according to how they define paracrine signaling, it is suppressing paracrine signaling. It's suppressing, you know, it's indirectly suggesting here thus far that it's suppressing it. And here are some of a typical slide, I guess. We'll assume it's typical. We have a control and treated. P21, stuff that's purple. DAPI is open arrows. DAPI is these solid arrowheads. I don't know, I guess they're missing arrows for P53 alone, but, you know, so here's your control. And over here, these arrows are pointing to the cells that are, so basically you're seeing less purple dots, right? So if you see less P21 being present, which I think is the case here, but again, you got to quantify this. This is just a little, you know, this is a qualitative picture. If you see less of the little pink ones here, then that should be less. So these aren't RFP. RFP is here. These P21s are your cells that are being indirectly paracrinely induced by surrounding cells. And if you have less of them, then I guess the SB525334 is you know, preventing those from senescing, or at least preventing those. Technically, more specifically, they're preventing those cells from upregulating P21 and thus not entering cell cycle arrest. Okay. Now we get to figure 4 and figure 5 and figure 6. And really, I'm going to stop sharing. Feels like this paper is taking a lot longer to go through than other papers, huh? I think I've just gone through too many figures, maybe. But those figures kind of laid the groundwork for figure 4, 5, and 6. And figure 4, 5, and 6 is where they actually start to, you know, test the compounds and see what happens when they, you know, when they add these compounds, and do these compounds work? I mean, the bottom line is, according to this paper, they do work. To me, you know, it's sort of like bearing the lead. You got the real cool stuff all the way at the end here. I'll stop sharing the screen. I'm just going to, you know, in case we have a power outage, I'm going to just scroll down to, and then I'll scroll back up, and I'll scroll down to figure 6, the very last figure. And here we have survival curves for mice, right? What happens when you add this other compound, AZ126011011? That's a droid that works for the Empire. No, okay, I got to stop. It's a chemical compound that's a soluble molecule that's fed to mice. That's basically an inhibitor of TGF-beta receptor 1, and they give it to the mice after a treatment after poisoning with acetaminophen, they basically do survival curves. So I think that it's a pretty large cohort of mice, 14 mice, and this is pretty damn impressive. I mean, here's your vehicle, which is probably whatever this stuff is dissolved in, and it looks like there's a couple of hangers on here, but man, most mice just drop dead right after like less than a day. And this stuff here is keeping them alive after like a week. I don't know if this stuff is available, and I just accidentally downed a bottle of Tylenol. I hope this is inside my medicine cabinet, is what I'm saying. Maybe they should have that. Maybe you should get a 2-pack when you go to a grocery store. There should be a bottle of Tylenol and a bottle of AZ126011011 next to that bottle.
We'll work on the name, yeah? But we preferably want something that doesn't sound like a protocol droid from Cloud City. But yeah, we'll get back to you on the name. But yeah, that's not really going to work.
Yeah. Anyway, I'm going to scroll back up here, and okay. So what are they doing here? Well, here they're showing what happens. Again, this is well, let me read the figure 4. TGF-beta signaling is activated. So here they're showing that the signaling itself is activated in acetaminophen-induced hepatocellular senescence. Again, these are from humans, so we're jumping into humans, and then they do mouse serial liver section. So the stuff in E down here is from mice. The stuff up here is from humans. And PSMAD23 is basically a downstream of TGF-beta 1 receptor. So it's what TGF-beta receptor turns on. It's a transcriptional factor. And P21 is what is also turned on. And you see that in normal liver, you just got DAPI, and then you got all of this stuff here, P21 and DAPI, and fulminant hepatic failure, which is secondary to acetaminophen overdose. So this is what happened. So yeah, if you overdose on acetaminophen, maybe you'll end up in a paper. No, that was a very crude joke. But anyway, yeah, it's a dark subject.
It's a quiet day at the office today, right?
Yeah. Let me move this. Okay, so TGF-beta expression, so control acetaminophen. So here they're staining for TGF-beta 1 expression. Again, you got to quantify these dots, and this area here is an area of necrosis, or actually no, it's the central vein area, and supposedly in the periphery is where you get this expression. So here they basically could quantify TGF-beta 1, I guess, versus total liver protein, baseline acetaminophen, TGF-1 beta expression goes up. So these are from liver extracts. SMAD7 probe copies. So basically they're using some sort of probe for SMAD7. Could be an RNA hybridization. I have to read but basically SMAD7 levels go up. A bit of noise here statistically. And here they see these. Again, this is really qualitative. Maybe they have somewhere, okay, this shows that there's P21, TGF, they have arrows pointing to this immunohistochemistry, but really without seeing the massive numbers there. And I'm assuming that this is higher than normal, right? So they give us this figure, and they kind of throw that in there. You know, I could kind of take it or leave it, this figure here. I mean, they're showing that this happens in mice too, but again, I'd like to, you know, like to see a quantification of what the heck I'm looking at here, and maybe be nice to see a control, basically non-acetaminophen treated mice. What are the normal levels of P21 TGF-beta receptor and TGF-beta 1? But anyway, okay, so really in figure 5 and 6 is where they start the treatment. What happens? And figure 5 is cool because they implicate macrophages. So macrophages release TGF-beta ligands, and they specifically here try knocking out these macrophages. And they lock them out by using these things called liposomal chlordronate, which I look lipclod. Can you come up with a better name? Lipclod? Okay. Lipclod. I guess it's got a better ring to it than
it's still not great. Where do they come up with these names?
Jesus, than SB525334. But anyway, lipclod. Yeah, I haven't had enough coffee. Sorry, I just had to suppress some. It's basically a liposome. So macrophages eat things. Liposomes are essentially these vesicles. They have this these soluble substance called chlordronates, which don't get through membranes, but they can get inside. And somehow I have to read up, but there's a company that makes these, and they're basically once they're ingested, this chlordronate stuff gets into the cell and is toxic. So by itself, it's not toxic, but once it's packaged in these liposomes and it's taken up by macrophages, it kills the macrophages. Yeah, lipclod. Anyway, so they want to see what happens since they basically are implicating in this figure macrophages, right? That macrophages, if there's damage, they get to the sites of damage. They're responsible. You know, all these things are good, but overdrive, they're bad. So they'll induce, you know, they'll cause inflammation, and they want to see if getting rid of macrophages might do the trick. And you know, the bottom line is that getting rid of macrophages seems to suppress this non-cell autonomous P21 expression. Okay, so here they have, again, immunohistochemical cross-sections, uninjured mice, which is good to see, and acetaminophen. CCL2 is a marker for macrophages, so CCL2 expression. So this brown stuff, I guess, is CCL2. So I guess this is staining macrophages. Again, it's really hard to see. I'd like to see also overlays of actual cell images, because you've got these little brown freckles here, and this is, I guess, corresponding to cells or inside of cells. So this is supposed to be macrophages, I'm assuming. So that's what CCL2 expression refers to. Acetaminophen F480 is also a macrophage marker. Let me just double check. CCL2, I think, is a macrophage. Yeah, it's a chemokine, staining expression of CCL2 chemokine, which is released by macrophages. And F480 is a marker for macrophages themselves. So you have nuclei, you have a lot of macrophages, and TGF-beta being kind of speckled around this area here. This other figure here, what do we have here? P21. So P21 expression, F480 and DAPI. So P21 expression going up. So we've seen these plots already in earlier figures, but here they count the numbers of monocytes, and how do they do the monocyte counting here in the area? Quantification of peripheral monocytes in mice. So I guess they count the monocytes literally microscopically. K per microliter, or is this in the blood? That's suggesting that it's probably a blood measurement rather than in the liver per se. I've got to dig in there a little deeper. But anyway, the monocyte level peaks one day after acetaminophen poisoning. And then they look at so let's see here, CCL2 inhibitor for 4 days. So they partially delete MDM2. So basically you have more cells that are p53 positive. So you have senescence induction taking place. And what do they add here? They add a CCL2 inhibitor, which I guess an inhibition for monocytes. So even though you have cells that are in the liver, hepatocytes and non-hepatocytes that are being senescent, you are also now inhibiting, I guess, kind of blocking CCL2, which is sort of blocking the macrophages, right, and seeing what happens. And non-cell autonomous P21 levels, which is indicative of paracrine senescence control. So CCL2 inhibition seems to be kind of blocking these levels of or blocking numbers of P21 positive cells, right? And BRDU is essentially, so here you have an inverse effect. You have more cells replicating, right? So adding in this inhibitor to monocytes, again, you know, in their figure here, it's implicating these monocytes in P21 induction. So they do some additional experiments down here with monocytes. So this is where they do add liposomal chlorhydrinate, LipClod, so partial MDM2 HEP, and so you have inductions of senescence, but then a few days later, you basically start clearing out the monocytes. So here they're inhibiting them, here they're basically killing them, right? And you're seeing kind of similar levels of relative P21 expression levels go down. And this is a field of view, I believe. Relative TGF-beta gene expression levels, so control, liposomal chlorhydrinate. I think this is, let me go through the figure here, F. Do they look at the entire liposomal depletion of whole mouse liver? So they quantified it by reverse transcription polymerase chain reaction. So basically expression levels of TGF-beta 1, 2, and 3. And that's pretty dramatic, that you have kind of this drastic inhibition of TGF-beta gene expression by essentially, you know, ablating your monocytes. Relative P21 levels go down, non-cell autonomous P21 levels go down, and BRDU positive hepatocytes basically go up. And this is another pre-recombinase system that they're using, and I think this is a different system, lyse and McCree. So this is a pre-recombinase that's knocking out TGF-beta 1. I think we've seen this before, right? You flux out TGF-beta 1, and obviously you're going to get more EDRU incorporation. Why are they using this system here? Let me take a look at G. Oops, sorry about that. Mouse liver mates, G. Let me go up here. There's figure 5G. There's the 5E, hepatocellular. Functionally test the role of macrophage-derived TGF-1-beta ligand. We use myeloid, so myeloid-specific TGF-beta deletion. Okay, in this case, they're deleting TGF-beta 1 in basically macrophages specifically, and they're seeing so you're not deleting these inside hepatocytes, but in macrophages, and you get more basically regeneration is what they're showing, right? So this figure here is implicating macrophages along with their TGF-1 beta production. But the bottom line is that, you know, regardless of the TGF-1 beta, whether it's coming mainly from the macrophages or also from the hepatocytes, blocking TGF-1 beta from binding to TGF-1 beta receptor seems to do the trick, as we'll see in figure 6. So if we go to figure 6 here, and this is where I zoomed in earlier to this mouse model, really, you know, all of this is for naught if you don't see something like figure 6b, which is the mice survive, right? So, you know, really, to me, that's going to be kind of the central figure. And if you add this chemical and they survive, then you can try to figure out how it's working. You know, for patients, really, that's the most important thing. And the cool thing is that knowing that it's most likely going to be through TGF-beta signaling means that there's all sorts of molecules you could now look at with varying degrees of secondary toxicities to inhibit TGF-beta signaling, right? And maybe there's lots of other molecules out there that don't just inhibit TGF-beta receptor, but also downstream components like SMAD and so on and so forth. So there's potentially a lot of interventions you can utilize, and a lot of them off the shelf, which is pretty cool, like this AZ12601000, whatever, that can be used to basically stave off liver toxicity. So here's the endpoint now. So I'm not going to go into this endpoint analysis, 16 to 18 hours. They mentioned here that in some studies they terminated this at 16 to 18 hours for humane reasons for the mice. But this graph here is clearly showing multiple days. So after you give Tylenol to mice, that's a pretty steep drop off, and that looks really, really good, right? That you have this much of a difference with a p-value of 0.0001 vehicle versus this. Serum bilirubin, this is a bit kind of, a little overlap and messy, but this is also indicative of liver damage, right? Because these are all molecules that are found in the liver, and if they're released into the bloodstream, then your liver is being damaged. So these are ways to measure liver damage. So that goes down the serum bilirubin levels with this compound AZ1260101. And here they use a different compound, SB52334. And again, you have basically similar effects, serum bilirubin levels. So this is not adding, this is before adding this compound. This is a vehicle control. So I don't know why they have it just 12 hours for pre, and then everything else is 2 days, 2 days, 4 days, 4 days. But anyway, so 2 days after acetaminophen addition, what is this vehicle control? There's really not much of a difference here. But at 4 days later, you see basically, the liver is getting damaged, and perhaps repair is taking place here because you see, really it says level of detection. So almost undetectable levels or normal levels of serum bilirubin compared to 4 days later, you see mice all over the place with these serum bilirubin levels. And then P21 hepatocytes per field go down. Just scrolling here. I believe this is more of the same. So I'm not going to stress this point because I'm kind of getting tired mentioning these numbers and letters. But basically the bottom line is that it appears that inhibiting TGF-beta signaling in cases of acute liver toxicity will promote liver regeneration and survival. And if that's the case, then that's pretty powerful stuff by inhibiting senescence. And to me, it would be really nice to see a nice chart. And I know Keith, our president, loves these charts, but I think it would be a really nice table to see, and we probably have a table. I don't know, I haven't looked in the Google Drive, Google Docs folder, but treatments that are being worked on now that have secondary disease applications other than aging that can expedite these drugs hitting the marketplace. And I think this is yet another example of drugs that can be used for acute liver toxicity that will have a spillover effect to as an anti-aging therapeutic, along with senolytics, which are being now used, the indication being increased senescence and organ failure due to basically poisoning from chemotherapeutics as a secondary, as a side effect. So I think it would be really cool if there's not a table like that out there, to have such a table. to me, I think I'd love to see something like these compounds on the market immediately, because, you know, unlike chemotherapeutics where, you know, they are toxic, but toxic over kind of a longer term. I mean, this is pretty as short term as it can get, right? I mean, if you have acute, you know, hepatic liver failure, you're going to be done before the week is done. Before the week's done, right? You're not making it to the weekend. So I would be surprised if we don't see, you know, experimental treatments being rushed down into the field to try to save lives, especially with drugs that are already been vetted through phase 1 and phase 2 testing. I mean, because literally, what do you have to lose, right? Nothing at that point.
Yeah, and that strikes the point that I was making a few weeks ago about the FDA not approving aging as a suitable endpoint for clinical trials.
Right.
The point, like you've raised just now, is these things are being used for other indications, other conditions, but then that means that the toolkit is there eventually because they get through the regulatory process, as I call it, via the back door. Look at Unity, classic example. Mm-hmm. They're going after osteoarthritis, quite a lot of other age-related diseases, and that means they get through, they get approved, and then we've got the tools, and then it's off-label use, isn't it? And that's going to be a hell of a lot quicker than, you know, trying to convince the FDA that aging is a thing, that, you know, is a disease. You know, obviously people are trying. I think you're probably going to get more traction nominating the hallmarks specifically as a syndrome, but that's another conversation entirely.
Yep.
Yeah, I honestly think the therapies and things, the toolkit, will be delivered via the back door. Like, for example, Mitosens. I think you'll see the application first with Mitosens in mitochondrial diseases, and they'll demonstrate the feasibility of, or not. I mean, obviously, I'm trying to be positive, but they hopefully will be able to demonstrate the feasibility of mitochondrial gene transfer and the techniques that they're suggesting in non-aging specific diseases. But then again, the technique then gets approved, and it can be then used for aging down the line. And I think it's the same here as you've touched upon.
Yep, definitely.
So don't worry, folks. Even if we can't quite get aging described as a disease in the ICD straight away, it's not going to stop. It's not going to stop people developing therapies, because we can treat aging-related diseases. And there's nothing stopping us to do that, or targeting the mechanisms.
So anyway, I just had a thought. So I wonder how much of an impact we have on the shares of Tylenol if we tell everybody to dump their Tylenol down the toilet. Will it really do we have that much of an effect? I don't know.
I don't think dumping Tylenol down the toilet is a good idea.
Hey, actually, no. Actually, it does. Yeah, scratch that. You don't want to be contaminating the water supply with Tylenol. So just try not to take it.
Yeah, it's a load of crap anyway. So it don't work, not really. And there are better alternatives.
Yeah.
So there we go. So conclusion: the liver is a highly regenerative organ as it is. So, you know, I'm curious to see how it would affect a relatively healthy liver, and could we?
Yeah, sorry.
And could also inhibition of TGF-beta work for other organs, for example? The work from 2015 by the Conboys suggests that it might. Certainly in mice, and TGF-beta is a common mechanism to us both, because you're going all the way upstream, all the way very close to the core processes. So, and it seems to work in much the same way. So could we regenerate? What I'm basically asking is, could I be Wolverine? Just this once?
Why not?
Oh, thanks, man. But yeah, I mean, you know, but in all seriousness, if we could work out what the factors are, and Irina suggests that there are only a limited number of them that effectively are involved in aging, then we could be looking at solutions not only for age-related diseases, but also, as you suggest, for things like liver failure and other traumatic injuries, where it's absolutely essential to upregulate regeneration as soon as possible. So, it could have a broad range of applications. And again, that also is another way of getting things through the back door, folks.
Yep. I just had a thought. It's probably people know this, but why are certain substances like acetaminophen particularly injurious to the liver, but not other organs? I know the liver, I know more or less what the liver does, and then it processes chemicals and then detoxifies them, and then you have toxic side products. But why, why the hepatocyte specifically?
Well, I mean, the organ is first in line, isn't it, really? It's the first thing.
It is first in line, but is it? Yeah.
What you would need to do is test tissue that hasn't been filtered by the liver, and then see if you get a similar result. You'd have to expose, I don't know.
Is it toxic to neurons, for example?
Or is it or isn't it? Or is it relatively safe once it's been filtered through by the liver? What's the liver removing?
Yeah.
You just don't know. So your liver is your first, last, and only line of defense against a lot of stuff, actually, which I suppose is why evolution saw fit to have it have a relatively higher regenerative rate than the rest of your organs in your body. It really is your sort of.
But I'm just thinking also from an aging research perspective, right? You take acetaminophen, liver has tremendous regenerative potential. Hepatocytes basically can divide like crazy, so they're very proliferative. So it'd be an interesting organ to study from like the somatic stem cell perspective and kind of the plasticity of the liver in general. But acetaminophen seems to destroy that plasticity, right? And it would be interesting to study it just from what's happening kind of at a basic aging senescence perspective, using that maybe as a kind of a tool. Why does it get, and I think that in the paper they even mentioned is very poorly kind of understood why there's this tipping point where there's too much damage and it can't regenerate. And I'm just kind of interested more fundamentally also at that molecular level of why it's too much for the liver to handle at a certain point, and what exactly is it doing to kind of squelch this regeneration, and certainly the induction. This paper suggests strongly that the TGF-beta signaling is implicated in this. So.
Yeah, I wouldn't be surprised, because TGF-beta is central in inhibiting stem cell mobility, and as demonstrated by Conboy, the Conboys, they showed that essentially once you expose beyond a certain level, obviously going back to the previous comment about balance, you need the right amount of TGF-beta, because if you don't have enough TGF-beta, bad things start to happen, right? Yeah. But if let's just say, for argument's sake, you have a youthful, or calibrate to a youthful level of TGF-beta, which is what they were saying as a possible therapeutic approach, then they found that the stem cell mobility increased and tissue regeneration resumed once they used ALK5 to inhibit it. Not totally, but what they did was knock it down to a level. You don't want full inhibition, because you need TGF-beta. You just don't need too much. But they think there's maybe 5, 6, or 7 sort of key signaling cytokines or chemokines that literally run the show, which is quite tantalizing, because it suggests that whilst aging is extremely complicated from a metabolic point of view, it's all sort of handled by a much smaller number of factors, if that makes sense.
Mm-hmm.
And that's quite you know, that's quite comforting to hear them say that we think it's only five, six, maybe seven factors involved in signaling, and it's a case of calibrating those factors, and they think that it could then spur systemic rejuvenation. And in fact, to slightly diverge here, what they're thinking about doing is using a technique similar to kidney dialysis, where literally you pass your blood through like various membranes, and it would filter TGF-beta, IL-6, and all these other, I won't say pro-aging factors because they're pro or anti-aging depending on their context. But what it would seek to do is it would seek to filter it or scrub your blood to the point where these things are balanced to a more youthful level and return it to you. Probably,
it's sort of a different spin on parabiosis.
It is, and it's they did some of the earliest parabiosis work back in the 90s, and of course they I think they taught Amy Wages the techniques, and they've had a lot of because they used to be in the lab next door, I understand, at one point. And who was another person as well? Was it I'm trying to think who it is. Not Rando. I can't remember. They were all in the same building at some point, and they've all sort of cross-pollinated. It's quite interesting. And yeah, I'll come to that name in a minute. I'll think of that name. Is it Rice? Weiss?
I can't remember.
But anyway, yeah, they but their proposal was quite simple, and it's something that's actionable, fairly realistic in the near future, because they're using a technique. I think it's called plasmapheresis. Plasmapheresis. That's it. Which is very much like the kidney dialysis that most people would be familiar with. It's a little bit more sophisticated, but they're working on the next generation version of this, which allows them then to calibrate these. I'm not going to say pro-aging factors, but these factors. So, and then thus return your blood, literally clean your blood, your aged blood, and pass your own blood back to you in a more youthful profile.
It's like changing the oil in your car.
It's like changing the oil, yeah, and of course it's one of the hallmarks of aging, which is the changes to.
You remember? So do you remember? You probably read this a long time ago. Fahrenheit 451.
Vaguely.
Well, if there's a scene in that book where his wife overdoses on something and the paramedics show up. So this book was written probably in the 1960s, I think, maybe 50s, by Ray Bradbury. Was it Ray Bradbury? Or was it, I think it was Brad. I have Google in front of me, but whatever. It's, and the paramedics show up, and what they do is they basically clean her blood by passing it through some machine. And basically get rid of the toxins that way. They basically give. So it's interesting that you mentioned this technology, because because for some reason that sticks out in my head from Fahrenheit 451. I just remember that scene in that book. It's science fiction. It's you could. It's a wealth of stuff in there that
sometimes we still haven't got those doors that go. We've got doors that open and close, but they don't make that noise.
Yeah, we're working on it.
We've got people. We've got our best people on that. Never mind about aging. We want the doors
make a swooshing sound.
Yeah, this is it. I reckon it was just somebody off the screen making a noise.
Well, I'm a biologist, so what I would suggest is you train a parakeet to make that sound. You just put it right by the door, so every time you walk next to it, the parakeet goes whoosh, and there's your answer.
Yeah, it's definitely an example of a near future plausible actionable therapy that we could take. The same for this. This approach with the liver. And I would imagine if you actually did have somebody who had liver failure, like we were talking about, and you put them through this scrubber, I'm going to use the word blood scrubber, would that even help address the issue as well by reducing TGF-beta globally to proper levels? it's interesting. And again, that offers another, potential route to going through the back door again.
That's true, and it's interesting to see what happens to tissues throughout the body if the liver is turning out, or TGF-beta into the bloodstream globally, but or damage in general. But anyway, much to discuss.
Yeah. So, and Irina Comboy's just joined our scientific advisory board.
Yes.
So,
welcome aboard.
Maybe as a thought, I know she was interested in doing a journal club.
Oh.
Maybe we can sort of have a look and get her in and talk about her 2015 paper, and let's talk tissue regeneration.
Yeah, let's do that. We've been talking about this a long time, getting SAB members on our journal clubs, and it's high time we did so. So perhaps we should go through our list of papers we'd like to discuss and things, and just pass them along to various SAB members and say, what do you think? What are some dates? And I think that would be very enlightening.
Yeah, I think so. I'd like to see a tissue regeneration special. That would work for me. So yeah, let's do that.
Okay.
So there we are. There's the conclusion. It's impressive stuff. Too much TGF-beta is bad, right? And
too much Tylenol is bad.
And too much Tylenol is bad. And this therapeutic approach could be potentially used for aging, but also things like overdoses of Tylenol and other issues with the liver. So yeah, it is something that's definitely worth exploring. Hopefully we'll not have to wait 4 or 5 years for a solution, and let's keep on keeping on.
Okay.
So that's great. So hopefully everybody enjoyed the show today, and we'll be back again next month. And thanks to all of our monthly patrons, the Lifespan Heroes. And did you know that we're almost at 200 heroes now as well? So, which is great going. And today we also won the Great Nonprofits 2018 top nonprofits list again.
Really?
We did, and we won the one last year as well. So that's 2 years running.
Excellent.
We're going for the hat trick next year, and that's great. So thanks very much, and to our patrons for supporting us. It means that we can make streams like this and do all the wonderful things that we do, and we really appreciate it. And hopefully we'll all see you all next time. And thanks for tuning in.
Yep.
And don't forget to check us out at www.lifespan.io/hero.
And everybody enjoy the rest of your summer.
Yeah, all 3 days of it, because it's gone really autumnal now. So
it's still hot here. So, but anyway.
Yeah, I think we got a few hot days, but not much. But there you go. Anyway,
all right.
We'll catch you all next time, and thanks for tuning in.